In the previous video, you got an overview of the cell division cycle, and that overview put the cell division cycle in a context perhaps different than the one you learned in genetics, where the cell division cycle was comprised of mitosis and interphase. In cell biology, we learned that the cell division cycle is a bit more complex. Yes, there is an M phase or a mitotic phase of the cell division cycle, but interphase is comprised of three different phases. The G1 phase, where the cell grows and undergoes normal metabolic activity, so this is a cell just being a cell. The S phase, where DNA is replicated, and a second growth phase, where a cell is preparing to enter mitosis. In the previous video, we learned that the molecules that regulate progression through that cell division cycle are complexes of a cyclin-dependent kinase and a cyclin. The identification that those proteins were responsible for driving mitosis was discerned through this experiment, where scientists injected the cytoplasm from a dividing cell, a cell that was in mitosis, into a non-dividing cell. That non-dividing cell was driven into mitosis. In other words, injecting the cytoplasm from a mitotic cell into a non-mitotic cell was alone responsible for inducing mitosis in that non-dividing cell. In order to control for the possibility that it was simply the act of sticking a needle into the non-dividing cell, or instead injecting cytoplasm from a foreign cell, to control for the possibilities that those were responsible for driving mitosis, the scientists did just that. They injected cytoplasm from an interphase cell into a non-dividing cell and saw that the non-dividing cell had no response. It did not enter mitosis. So that confirmed that there was something in the cytoplasm of that dividing cell, the cytoplasm of that mitotic cell, that was responsible for driving the non-dividing cell into mitosis. That something was called MPF, and that stood for mitosis promoting factor. Mitosis promoting factor, or MPF activity, is highest during the process of mitosis. And indeed, there is no MPF activity during interphase. This somewhat contradicts the previous video where the speaker implied that MPF activity was increasing during the S and G2 phases of the cell division cycle, but that's not true. It in fact rapidly increases at the start of mitosis and rapidly decreases at the end of mitosis. As we said, now we know that MPF is an activity that is comprised of two proteins, a cyclin-dependent kinase that is present throughout the cell division cycle that complexes with a cyclin, and the cyclin protein levels rise and fall at different portions of the cell division cycle. So M-cyclin, that is responsible for activating MCDK, slowly increases to the point where it peaks in mitosis. Where the concentration of M-cyclin peaks, MCDK activity is greatest, and again, that is during mitosis. M-cyclin paired with MCDK is not the only cyclin/cyclin-dependent kinase that we need to consider when we're looking at the cell division cycle. Indeed, there are several cyclin/cyclin-dependent kinase complexes that are active and have peak activity at various stages of the cell division cycle. M-cyclin is shown here, but so is S-cyclin and S-CDK, whose activity peaks during the S phase of the cell division cycle. Importantly, the previous video spoke about the checkpoints along the cell division cycle. What drives a cell past each of those checkpoints is the relative concentrations of the various cyclin/cyclin-dependent kinase complexes along the continuum of the cell division cycle. The previous video spoke about a checkpoint in G1. That checkpoint in G1 is referred to as the restriction point. Sometimes it's referred to as the start point of the cell division cycle. And that is because if a cell passes through that restriction point, passes through that checkpoint, passes through that start, it will proceed through the rest of the cell division cycle. The only other option for it, if it is unable to pass through all of the stages of the cell division cycle, is to undergo a process of programmed cell death. So bottom line, if a cell passes through this R restriction checkpoint, it will either complete cell division or it will die. As I said, it's the ratio, the relative ratios of the cyclin, of which cyclin/cyclin-dependent kinase complex is active that will allow us to pass through each of the checkpoints along the cell division cycle. The relative ratios of cyclin D coupled to G1 cyclin-dependent kinase and cyclin E coupled to G1S cyclin-dependent kinase, the relative ratios of these two complexes that will allow a cell to pass through that restriction point. Here we see a graph showing the concentrations of each of the cyclins through the various stages of the cell division cycle. We see that cyclin D slowly increases through the G1 phase, and at some point in G1, cyclin E begins to increase substantially, and at some point that concentration of cyclin E exceeds that of cyclin D. That also suggests that the activity of the G1 cyclin-dependent kinase is exceeded by the activity of the G1S cyclin-dependent kinase. It's at that point that the restriction point, or that checkpoint, is surpassed. Again, we're passing through that checkpoint when the cyclin E concentration becomes sufficient enough to activate G1S cyclin-dependent kinase above that of the G1 cyclin-dependent kinase. When we pass through that restriction point, the cell progresses through the rest of the stages of the cell division cycle. If the concentrations of cyclin E and the activity of the G1S cyclin-dependent kinase never exceed those of cyclin D G1 cyclin-dependent kinase, then a cell will enter that G0. G0 is a sort of rest stop, if you will, along the cell division cycle, where a cell will wait indefinitely, if necessary, until that cyclin E concentration causes the G1S cyclin-dependent kinase to rapidly increase.